Okay, hello everybody. Okay, so we are going to talk a little bit more about skeletal muscle today. We do have an exam on Monday, so anything from today will be limited in number of questions and will be a little more general rather than super detailed. But I want to make sure that we're getting through content so that as we have like breaks, Thanksgiving break coming up, we can take things a little bit easier around then and not have to jam pack too much in our final. Okay, so when we left off, we talked through the cross-bridge cycle. So last time we did sarcomere cross-bridge cycle. I posted a sarcomere activity as our quiz for this weekend. You can keep taking it over. Comes with a set of slides that you can kind of drag around so that if you're having any trouble with sarcomere structure, you got a couple more ways to look at that. But let's go over one more time, one little review question on the cross-bridge cycle, because this is important. Let's take a look. Okay, so I'll pull up the cross-bridge cycle in a sec to go over this. But yeah, what we're doing with the ATP is we're using it to break apart our actin and myosin and taking the energy out of that bond between our final phosphate groups to put myosin into a high energy form, aka one that can do this cross-bridge cycle over again. Okay, so what we're looking at for this question is like this part up at the top, right? So we can see that here, when the ATP comes in and binds to the myosin, that's this fourth step where our myosin and actin break apart. And then the other thing that ATP is doing is we break it up. And so we can see in step 5, we pulled that myosin head back in. We can see that the energy is now in the myosin in this high energy form. And now ATP, which was higher energy, is turned into ADP and a phosphate group. All right, so we're kind of like repositioning it so that we can do this over again, bind myosin to actin, and have room to move the myosin and do this power stroke, drive actin. When we get to different types of muscle fibers, which we will do before going into the heart in our lax unit, we're going to see the different types of muscles process ATP a little bit differently. So that's a point to remember. Okay. But for today, we want to link up what we talked about with our neuromuscular junction and the somatic nerves to what we now know about how our muscle fibers contract using this cross-bridge cycle, shrinking our sarcomeres. Okay. So when we talk about excitation, contraction, coupling, all we're saying is that we have a sequence of events that takes us from the end plate potential to propagating an action potential, cross the sarcoplasm down into the muscle fiber and releasing calcium to expose these myosin binding sites on the actin. So calcium wasn't in the cross-bridge cycle in terms of its steps, but we'll now see this little green piece on top here. That's our calcium ion. So that's bound onto troponin already in this cross-bridge cycle. Okay. So what this depends on, right, is the neural input, right? So the action potential coming down our somatic nerve from that motor neuron. And then we're releasing calcium from our sarcoplasmic reticulum so that our actin has that binding site for myosin to grab onto. Okay. So we got steps. So we're going to start off where we left off with neuromuscular junction. So we left off, we had released acetylcholine, we had a nicotinic cholinergic receptor on the motor end plate that responds to that acetylcholine by letting ions flow. On the whole, we have lots of sodium come in, lots of cations come in any case, meaning we depolarize a bit. We have this end plate potential, right? So we have a membrane potential that's rising. So we are exciting the cell at this point. Right? So now we have that end plate potential growing into an actual action potential. So this is a muscle version of an action potential. Across that sarcolemma, so across the plasma membrane, we have a membrane potential, right, is the difference in charge across the membrane. So that action potential is going to travel across the membrane, down the membrane, until it hits our T-tubules. So the T-tubules were part of that triad we had with the sarcoplasmic reticulum. So literally like tubes going down into the cell. Okay? And as that action potential travels down these tubes, we have receptors in the tubes that open calcium channels that have been waiting to release the stored calcium in the lateral sacs of the sarcoplasmic reticulum. So when we send this action potential down the T-tubule, the calcium gets released from that sarcoplasmic reticulum into our cytosol, so into the cytoplasm. And it's now free to bind onto troponin, which is what shifts the tropomyosin, that long thready pink thing, off the myosin binding sites on the actin, which means we can start our cross-bridge cycle. So we'll take a look at this in picture form too. It's our big thing. So we were starting where we left off. So this is our neuromuscular junction. We know we have different names than we did in our synapses in the central nervous system, but a similar process for this release of neurotransmitter. Okay, so here we have an end plate potential starting to form, and it's going to head down our sarcoplasmic reticulum, so down our membrane. Oops. Okay. So before we look at the rest of this picture, or while you have the picture of this whole process up, it's fine with me if you look for right now, want you to make sure that you can make sense of what some of these named structures were. So each one is the one that we were just saying moves the action potential down into the muscle fiber. Okay, yeah, that is the transverse tubule. Okay, so here we see that step, right? So our action potential formed out of our end plate potential here at the motor end plate, moving down the sarcolemmas, but this is just taking us down to the side, right? So we're moving kind of along the outside of the cell. And it's not until we get to the T-tubule that we run into the cell, right? So here's that T-tubule. So this is a structure we didn't see in our neurons. This is going to be specific to our muscle fibers here. Okay. So here's that T-tubule going down, and here's the sarcoplasmic reticulum with its lateral sacs, making that triad, right? So one T-tubule in the middle, sarcoplasmic reticulum to either side for a total of 3 structures right here. Okay, so we can see these little green dots inside that sarcoplasmic reticulum in our figure. Those are our stored calcium ions. And as we have this action potential travel down the T tubule, once we're in contact with that sarcoplasmic reticulum, we have receptors sensing that change in voltage. And the result is we release calcium into the cell. Okay. And that takes us to essentially our cross-bridge cycle, right? So to this point, right, our actin would have had its myosin binding sites covered. I'm not going to draw the whole thing, right? But we've got these olives. That center of the olive is where our myosin needs to bind on, right? And when we're not cross-bridge cycling, it's blocked by the tropomyosin. So what you can see that's different here in this fourth step is that tropomyosin has been pushed to the side, right? So now all of those myosin binding sites are uncovered, right? The tropomyosin didn't disappear, it's out of the way because the calcium has bound to our troponin complex, which is basically acting like a little switch that pushed the tropomyosin to the side. And then our familiar cross-bridge cycle. So at this point, we are going to start to see muscle fibers contracting because as soon as we expose these myosin binding sites on the actin, we're already set up, right? Like we already hydrolyzed some ATP. We've got myosin in its high energy form kind of ready to go. Now it has somewhere it can grab to, so it does. And that's how we kick off our cross-bridge cycle. Eventually, we're going to soak that calcium back up. You don't want to cross-bridge cycle forever. You don't always want your muscle cells to be super stiff and contracted. So over time, we do take that calcium and actively, so using energy, transport it back into the sarcoplasmic reticulum so that we can do it again later when we get another signal from another somatic. Big picture. Action potential coming down our somatic nerve, acetylcholine in our neuromuscular junction. Eventually we have an action potential on the sarcolemma of this muscle fiber, travels down through a T tubule, resulting in calcium release from our sarcoplasmic reticulum. And that's really it when we're thinking about this excitation contraction coupling. We're just talking about how do we get from the synapse-y seeming stuff to the contraction part. The answer is calcium. Okay. So eventually, if we soak back up the calcium, then we go back to our original position for the actin where those myosin binding sites are blocked. And at this point, your muscle fiber is going to relax. And here that is zoomed in its 2 forms, right? So here we see the relaxed with tropomyosin blocking those myosin binding sites, right? So that's our tropomyosin, the long skinny thing. Here's that troponin complex, a little snowman. And here's what happened when we bound on calcium. Right? So we pushed this over to the side. So we now have the tropomyosin shifted off our binding site. Okay. So to finish contracting, to relax your muscle, you need to remove the calcium from the troponin, and then you need to actually remove calcium from our cytosol, right? From our cytosol, from our cytoplasm, from our sarcoplasm, because if it's floating around there, it's going to bump into troponin again, and we're just going to keep trying to do this cross-bridge cycle, keep using ATP. But there's also only so short you can shrink a sarcomere. So there's really no point in doing this forever beyond the fact you don't want to just stay contracted all the time. So we have an enzyme that helps us with this. It happens to be called our calcium ATPase in the sarcoplasmic reticulum, or our SERCA. Okay? It's just pulling calcium back into the sarcoplasmic reticulum. So we can take a look at this coupling.
Typically, a single motor neuron arising in the brain or
time
spinal
it was
cord
quiet.
conducts action
Turn
potentials
super loud.
that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad.
We've got some variation in
The
terminology, but
membranes
that's
of
the
the
same
T-tubule
structure
and
as
terminal
our
cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
All right, so lots of words, but that's our whole big process. And if you zoom out a little, hopefully that makes it a little easier to remember. If you can actually do this with your own body. Okay, so next thing we're talking about is the twitch. So what we're talking about when we say someone twitches, when we say a muscle twitches, is we're talking about specifically what happens when one action potential comes down a motor neuron and then a muscle fiber, right? So this is going to be an all or nothing event, kind of like our action potentials in our neurons were all or nothing events. We're going to think about our twitch as happening or not happening to any given muscle fiber at rest. Now, where it does get confusing is that this term can be used at multiple levels depending on what people are focusing on. So we might say that we have a twitch in a muscle fiber. We might say that there's a twitch in a whole motor unit, or we might talk about the twitch happening at the level of like the whole named muscle, which has many motor units inside it. Each of those motor units has many muscle fibers inside it. But the concept is going to be kind of similar for all of them. We're going to do one more review question. Right, so we did mention motor units before. I want to make sure that we remember what those are, because in order to talk about how twitches work, we're going to want to know where it's happening. So I'm going to pull up a question in a second, but I want you to look at this picture. I want you to find how many motor units we are looking at. Okay, let's take votes. Oh, vote on it. No? Did you all vote, or are these old? Did anybody vote already? Okay, we got some mystery voters in here. Spooky. Okay. Okay, so it should be two. Let's look back at that picture. So remember what a motor unit is all the muscle fibers that go back to one motor neuron, right? So when we're grouping together muscle fibers into a motor unit, what we're noticing is that we have one neuron that then splits out to several muscle fibers, right? Which means that if you send an action potential down this somatic nerve A, on this motor neuron, all of those muscle fibers that are attached to it at the end are all going to get this action potential basically at the same time. So they're going to contract together, right? Because they're all responding to that same thing. So one motor neuron and several muscle fibers is our setup for a motor unit. Any given muscle fiber on the flip side is only going back to one of our somatic nerves, to one motor neuron. We don't have any fibers that are getting innervation from like both A and B, one or the other. Okay. All right. So when we're talking about a switch, we're going to think about it in phases. So think kind of like when we were looking at action potentials. Right? And our big goal is to understand contraction. So we're going to see that at first, we're going to have a bit of a latent period. So we're going to have a little bit of a timeline here from when that action potential goes down our muscle cell, goes down that muscle fiber, to when we actually see sarcomeres shortening, when we actually see contraction happening, right? So this is going to be about a few milliseconds. So this is the time when we're going to be doing that excitation contraction coupling we just talked about, right? So we're going to be sending that action potential down our T tubules and triggering that release of calcium from the sarcoplasmic reticulum during this time. So we have a couple steps that need to happen here. Then we're going to start to notice actual contraction, right? So the sarcomeres, the muscles actually getting shorter. The sarcomeres are definitely getting shorter. We'll see whether the named muscle as a whole gets shorter or not. It's going to depend on some things. But sarcomeres are certainly getting shorter during this contraction phase. So this is when, if you measured tension in a muscle, it would be building. So this is going to go on for 10 to 100-ish milliseconds here. And this is when cross-bridge cycling is happening. So this is when we have that myosin grabbing the actin over and over again, pulling it towards the center of the sarcomere, shortening that sarcomere. Then at the end of the twitch, got like the full twitch visible, we're going to have our relaxation phase. So we are going to be decreasing that tension in the muscle back to zero. This actually takes longer than it took to contract in the first place. Relaxing is actually kind of hard. Okay? So during this relaxation phase, this is when we should be taking our calcium back into the sarcoplasmic reticulum, so it's not swimming around in our cytoplasm trying to get us to keep cross-bridge cycling. Okay. So they look like this if we're looking at the force of our contraction, our tension, right? So here we have tension. Oops, really bad at circling stuff. Here's our tension on the y-axis, okay? And here's what we're looking at over time. So are we seeing tension build or are we seeing tension lessen? And here's our stimulus. So you want to think this is when our nerve signaled to our, we'll say muscle fiber is the level we're looking at. So here we have our brief latent period. So you can see that there's no change in tension for this first little chunk of time. All right, so we got our stimulus, but we're still doing all the steps that we need in order to get that cross-bridge cycling going. And here we have our contraction phase next in purple, right, to our peak tension, right? So this is variable in how long it takes, but relatively short compared to our relaxation phase, which we see spreading out to the end of this twitch, right? So it's taking us longer to relax as we try to soak up all that calcium that we release. Okay. think I'm gonna keep talking so we get through content. But this is the point at which I kind of planned for exam content to end. So remember calcium in our excitation contraction coupling, and I want you to know the phases of a twitch. Okay. Now I'm going to talk about our different types of twitches and some additional stuff, and we'll push that into a future quiz for assessing it. Okay, but do listen. Okay, because this is kind of interesting. It's something you can try on your own in your own bodies as we're trying to understand these twitches. Okay. So when we're thinking about what's actually happening during contraction, okay, our contractile elements are our sarcomeres, right? And by that, I mean, that's what's getting shorter. We know the actin doesn't get shorter. We know the myosin doesn't get shorter. Just as the sarcomeres get shorter because our actin and myosin slide past each other. Okay. Now, if you're looking at a muscle in anatomy lab, you're not looking at just one muscle fiber, right? You're looking at, I draw some terrible bones. Okay. All right, so you've got some bones. Let's say this is an arm. Okay, great. Okay. So when we're looking at the muscle, you have attachment points on our ear bones. And then you have a muscle belly between them. Okay. So the sarcomeres are in what we think of as the muscle belly, right? So that red part of the muscle. But the muscle as a whole also includes these endpoints, right? Which are going to be different types of connective tissue, but in particular, you're thinking like the tendons, right? You've seen those long, skinny white tendons. Those don't contain a bunch of sarcomeres, so the tendons themselves wouldn't be contracting. It would be the muscle belly and the muscle fibers that they're attached to. So when we're talking about a muscle with a name, like your biceps or something, we're talking about the whole thing, including both muscle fibers and these connective tissue elements. So the tension in the muscle is getting generated as that muscle belly is contracting because its sarcomeres are getting shorter. Okay. And you also have some force that you're working against, which we think of as our load, right? So easy to think of a load as if you think of lifting an object, right? The thing that you're lifting in your hand, that's your load here. But you also have a load if you're pushing against something, right? It's not when you're able to move, right? When I push against the wall, but you can still feel tension in the muscle, right? You can still feel that you're contracting the muscle. You're still doing something. Okay. And so that's going to be the difference between our two types of twitches, is this relationship between building tension and actually moving a load or not. Okay. So when we talk about an isometric twitch, or if you're talking about an isometric exercise, we're going to think about starting out with a muscle where this length is staying constant, right? Our contractile elements are going to contract. So actually, let's reverse this. Did I draw the muscle belly well? I hope. I'm going to redraw it. Okay. All right. So we're going to go from a little skinnier muscle belly in the sort of relaxed state to everything is going to start to bunch up, right? So as you contract, what we're going to end up with is something like stretched out tendons and then bunching in the center of our muscle, right? So the overall length in this situation, even though the muscle belly has changed, the overall length of the muscle, when we think from origin to insertion, so like from endpoint to endpoint, that length hasn't changed. And the reason for this is that we haven't exerted enough tension to actually move our load, right? So either you're trying to lift something that's way too heavy for you, or your load is something that you just like can't move, right? So instead, what's happening is that the elastic parts of a muscle, so not the contractile part, so like the tendons, right? These endpoints, right? These have stretched out, right? So they're making up for the fact that the muscle belly, where we have the sarcomeres, where we have those myofibrils, they are shortening. So we're making up for it. So our length doesn't change. Okay. So if you look at to measure this or what measuring this might look like, if we're looking at an isometric muscle contraction, as that twitch happens, right, we are going to see, here we've got our point where we're hanging our muscle from. Here we've got our force transducer. So this is what's measuring tension. Okay. Here's our stimulator. So we've got some like electric probes shocking this muscle to get it to contract. In the body, which would be a nerve. Okay. And we're taking a recording of what force did we generate or what tension did we generate here. So here we can see in the contracted state, right, that distance from endpoint to endpoint didn't change. But what this center part of the muscle looks like did, our tendons stretched out, our muscle belly, the actual contractile part bunched up. So over time, we could see the parts of our twitch on our tension graph. All right, so we'd start with our latent period. All right, so the part where we have nothing happening in terms of tension generated, but we know that this is where we're linking up our excitation to our contraction, so we're releasing calcium. And then we can see the actual contraction, right? So this is when the muscle belly bulked up, right? But we didn't actually move a load, and then we relax, right? So there's that longer relaxation phase. So that's what a graph of an isometric muscle contraction would look like. An isotonic twitch, if we record the tension, is going to look a little different. The reason it's going to look different is we're actually strong enough to move the load at this point, which changes how much tension we have to generate, basically. So in an isotonic twitch, instead of keeping the length of the muscle constant, we are going to keep the tension in the muscle constant when we get to this isotonic part. So our tension in an isotonic twitch is going to be greater than our load. So right now we actually can lift stuff, right? The load on my hands is pretty light. I can exert potentially a lot more tension with my muscles here. So we actually lift the load, so the whole muscle actually gets shorter. Because if it didn't, nothing would move. So here's what that looks like. Okay. So in this case, we can see that between our relaxed state and our contracted state, we've actually changed the distance now, right? So we had a long muscle here. We've got a shorter muscle overall when it's contracted. We can see that some of that is coming from the muscle belly, from the red part, but we also are letting the tendons move along with it instead of just stretching them out. Right? So we have enough tension that we built up to get our load to actually move here. So over time, right, we can see that our tension graph looks a little different than our initial picture of what a twitch looks like. Right, so we still see that we've got like a latent period, so we've got our time lag. We still have a phase where we're contracting and the tension is building, but then we have this plateau in the middle here, right? So this is the part that makes it actually isotonic, because what's happening there is that instead of building tension more and more and more, you don't need to exert more and more force once you've met the amount you need to actually move a load. So instead, we're going to see the tension kind of staying at that amount of strength, basically. And that plateau matches up to the time when the muscle is actually shortening, right? So you're keeping that constant tension, right, but shortening the muscle as you move the load. And then we see relaxation again, in this case happening at the end of that plateau after we've fully shortened our muscle. So when we think about twitches, we do have some twitches that are purely isometric, which is why that's the picture we use to understand the phases of a twitch, right? The isometric graph is what we saw when we were looking at generically naming them. But to get a purely isotonic contraction is a little harder because usually when you start lifting something, you don't immediately move the load the second you start contracting the muscle, right? Like your load has some mass to it, right? So we usually have to actually build up our tension for a little bit. So have an isometric part before we hit this amount of tension where we're actually able to move the load. Okay. So usually we have an isometric phase and then an isotonic phase if we're looking at the overall twitch of an isotonic exercise. Okay. Now, an interesting thing to be aware of, though we're not going to be drawing out lever arms and calculating how loads change, you might notice that loads in general aren't actually constant. If you lift something, right, you're trying to lift something, there are points at which that feels harder and there are points at which that feels easier. And that's because as your limb position changes, right, like how close the load is to your body versus how far, exactly what position that muscle happens to be in, that actually does change how much load you're working against in order to move something, move your muscle. So this is like, you can think about if you've ever moved a couch with those like moving straps, it's way easier than without moving straps, right? You're kind of artificially changing the position of your load, the couch, versus your body and your muscles. And that kind of happens all the time as you're actually physically moving an object. So when we look at muscle contractions overall, we do feel there's some gradation there, right? Like you can imagine strong contractions and weaker contractions, right? From your daily life, you're not always using your muscles at full strength. Right? Now, this is because we produce tension in each of our muscle fibers, but we don't always contract all of them every time we contract that muscle. We're actually going to see that we have different numbers of motor units kind of coming online to contract together as we realize we need more and more tension to move a heavier object. So in order to produce more tension, you want to recruit more muscle fibers, right? So more cross bridges in more muscle fibers means we have more force overall, right? Like more sarcomeres are shortened. So the more fibers you have contracting, the stronger you're going to see that muscle behaving. You're going to see more tension building. So when we see our force generated by individual muscle fibers, you can see that there are also some interesting relationships to successive stimuli, right? So if you stimulate a muscle over and over and over again, we're going to see some kind of warming up until we hit a plateau. And we're going to stop here and talk about some of these extra factors after our exam. We've got a couple more things coming together. I think it's a good place to leave it.